Assalamu alaikum, future doctors. Today, we are diving deep into the core of modern biotechnology, recombinant DNA technology, also known as genetic engineering. This topic is not just important, it's a fundamental concept that underpins almost everything else in the biotechnology chapter. Our goal is to understand this process so well that any MCQ on this topic becomes an easy score for you. We will cover four main areas. First, the core principle of recombinant DNA technology. Second, the essential molecular tools required. Third, the detailed step-by-step process. And finally, a classic application, the production of human insulin, also known as humulin. Part 1. The core principle. What are we actually doing? At its heart, recombinant DNA technology is the process of taking a specific gene from one organism and inserting it into another, usually a simpler organism like a bacterium. Why do we do this? The goal is to make the recipient organism produce the protein that the inserted gene codes for. We are essentially turning simple organisms like bacteria or yeast into tiny programmable factories for producing valuable proteins.
Dreaming of MBBS? I chose Stars Academy Lahore for MDCAT prep, and here's why. Chapter-wise tests with video discussions, quarter, half and full book tests, topic-wise tests.
Stars Academy Lahore is one of the best institutes for MDCAT preparation.
If you want to prepare for the MDCAT, I highly recommend Stars Academy Lahore for their excellent coaching and supportive environment.
Topic-wise tests with detailed explanations. Even past papers are explained chapter-wise. Quick revision? It's all online and super effective.
Let's understand some key terminology. Gene of interest, the specific gene we want to clone, such as the gene for human insulin. Vector, a delivery vehicle, typically a bacterial plasmid, used to carry the gene of interest into the host cell. Host organism, the cell that receives the new gene, usually E. coli bacteria or yeast. Recombinant DNA or rDNA, the resulting hybrid DNA molecule created by combining DNA from two different sources. For example, a human gene plus bacterial plasmid. Transgenic organism or genetically modified organism. GMO, the host organism after it has successfully taken up the recombinant DNA. Part 2. The essential molecular tools of the trade. To perform this cut and paste operation at a molecular level, we need a specific toolkit. For your MDCAT, you must know these three key tools. 1. Restriction enzymes, the molecular scissors. Function. These are enzymes that cut DNA at very specific sites. They are the scissors of genetic engineering. Specificity. Each restriction enzyme recognizes a specific, short nucleotide sequence called a restriction site or recognition sequence. These sequences are typically palindromic, meaning they read the same forwards and backwards on opposite strands. For example, GATTC, sticky ends, the most useful restriction enzymes, like EcoRI, make a staggered cut leaving short, single-stranded overhangs. These are called sticky ends because they can easily form hydrogen bonds with complementary sticky ends. This is crucial for pasting the gene into the vector. 2. DNA ligase, the molecular glue. Function. This enzyme is the glue after the gene of interest and the plasmid have been cut with the same restriction enzyme. Their sticky ends will pair up. DNA ligase then joins them permanently by forming strong phosphodiester bonds in the sugar phosphate backbone. 3. Plasmids, the vector. What they are: small, circular, extrachromosomal DNA molecules found in bacteria. Why they are used as vectors: they can be easily isolated from bacteria. They can be cut with restriction enzymes to insert a foreign gene. They can be readily taken up by host bacteria. They have an origin of replication, ORI, allowing them to replicate independently of the main bacterial chromosome, creating many copies of the gene of interest. They often contain marker genes, such as genes for antibiotic resistance, which help us identify which bacteria have successfully taken up the plasmid. Part 3. The step-by-step process of genetic engineering. Now, let's put it all together. Here is the complete workflow for creating a recombinant organism. Step 1. Isolation of the gene of interest and the vector. The gene of interest, for example, the human insulin gene, is isolated from human cells. The plasmid vector is isolated from bacteria, such as E. coli. Step 2. Cutting the DNA, restriction digestion. Crucially, both the gene of interest and the plasmid are cut with the same restriction enzyme. This is a vital point for MCQs. Using the same enzyme ensures that both pieces of DNA have complementary sticky ends. Step 3. ligation joining the DNA fragments. The cut gene of interest and the cut plasmids are mixed together. The complementary sticky ends pair up through hydrogen bonds. DNA ligase is added to the mixture. It seals the gaps by forming phosphodiester bonds, creating a stable recombinant DNA molecule.